Invitation and the introduction, and hello everyone. Let's see. Everything visible now?
Yes, we can see. Thank you.
Wonderful. All right. So when you reached out, you said, okay, well, let's talk about safeguarding society. And I thought, well, let's talk a little bit about where we're going as a society as generative AI gets really, really, really good. How can we ensure that we can reap as many of those benefits to build a flourishing society without anything going wrong and sparking some kind of backlash or something perhaps even worse? So I'm going to give a hopefully briefer talk than that because I would really love to hear many of your thoughts in a subsequent discussion. I'm going to begin by being somewhat controversial and invite everyone to push back if you have an objection to something that I say, because I think that's really important that we work these things out in open discussion. So I'm going to begin by saying why I'm a little bit concerned about extremely powerful biotechnology that is in everyone's hands to do whatever they want with. And that's just that if we think about a security framework, if you talk to folks who do a lot of cybersecurity or information security, and you think about biology as a field, well, it's characterized by its ability to replicate. Meaning, if something is highly fit in the environment, then it will spread if growth exceeds death. And an exponential technology, we'll call it, is one that can spread widely on its own without any human intervention. What's interesting about exponential technologies is that this gives them a great deal of power. So on the left, you can see a quick diagram of mosquitoes and a gene drive. One of our original publications showed that you can use CRISPR, you can encode CRISPR genome editing system into an organism along with an inserted gene that changes the phenotype of that organism. And in each generation, in a heterozygote, CRISPR will cut the original chromosome inherited from the wild type parent and replace it with itself and the edited gene. So that means that when this happens in the reproductive cells, if it works, all of the offspring, rather than only half, will inherit the edited gene. And if the trait is always inherited, that's almost twice as fit in heterozygotes, meaning it will spread through the population. Now, this is really neat if we want to do something like edit all the mosquitoes so they don't spread malaria. Or we can do other things. We can engineer them all so that they all develop as males. The population will crash. When you model it, it looks like the population won't go away. There will still be mosquitoes. It's just their population levels will be too low to sustain transmission of malaria. And we could do this for other things, for parasites. We could target schistosomes directly. Schistosomiasis is a terrible growth stunting disease, very common in many of the areas that malaria is present. We could use this to suppress populations of the New World screwworm, which is this horrible botfly that lays its eggs in open wounds, and the larvae drill their way into living flesh and release chemicals that summon volatiles that summon new gravid females to lay more eggs. So the animal gets devoured alive by flesh-eating maggots. Really nasty. We eliminated it from North America entirely by, believe it or not, radiation sterilizing millions of them and releasing them, and did this over decades to eliminate them from North America entirely. And we've kept them penned in South America for the last 15 years by releasing 10 million sterile screwworm flies over Panama every week. But recently, the flies actually broke through and now they're spreading back towards us. So we're going to have to stop that and push them back down. But South America would like to control them as well. Gene Drive could do that. Okay, that's great. But if you can engineer a mosquito so it doesn't spread disease, you could engineer a mosquito so that it does spread disease. And when I first thought we might be able to do this, I was a little wary of this. I decided to sit on it and think for a bit. Is this a technology that could be misused? Does it favor offense or defense? And I concluded that in order to favor defense, an exponential technology, something that can be potentially released by one person acting on their own, it has to be slow, so we have time to respond to it. It has to be obvious, so it doesn't sneak up on us. And it has to be easily blocked. We have to be able to do something about it when we see it and have time to respond. So a pandemic, we know, is, I would say, offense dominant. COVID was pretty fast in terms of spreading around the world. It was sort of obvious. You could see it, but it did the asymptomatic transmission to some extent. And our vaccine response was amazing, a world record, but it was also comparatively slow relative to the spread of the virus. We didn't have enough time to prepare defenses before it was everywhere. So that means we're pretty vulnerable to future pandemics. If someone knows how to cause a pandemic and can access a relevant virus, then they could cause a pandemic. And then we would be forced to respond by trying to develop a vaccine and going through the whole COVID experience again. Then there's something that some of you may have heard of. We had a paper out in December in which we describe what would likely happen if we were to make a mirror bacterium. So this is an organism where every chiral center in every biomolecule throughout the cell is flipped to be the mirror image version, to be the enantiomer. And because it looks like virtually every form of immune recognition and lots of immune killing and predator recognition and predator consumption are overwhelmingly based on chiral recognition, it looks like by default, mirror bacteria would be highly immune evasive and have quite a few fitness advantages. Now, this is highly uncertain. And one of the things I really hope science does over the next 6 months or so is think about whether or not this diagnosis is incorrect. We really need to challenge it. But right now, it looks like it would spread widely. It would spread into us. Our immune systems would not be able to respond effectively. It would spread through the food chain. There would not be predators. It could plausibly infect plants and animals and lots of things. And it would probably spread pretty fast. And there wouldn't be really any clear way that we could do about it, at least not without a lot of preparation. We could almost certainly save ourselves and a few species that we want if we engineered correctly using some amazing new technologies like sniper receptors and so forth. But it would be bad for the world. And so that's the sort of thing we just need to not develop, I personally think. But again, we could be wrong. And I would love to have folks weigh in on that one. Let's go back to pandemics, because mirror life is thankfully at least a decade away and hopefully much more than that now. So pandemics, though, if you look at the number of leaders of nuclear armed states, you're at slightly above 10, and it's been that way for quite some time. But as soon as we reconstructed the 1918 pandemic influenza virus, the number of people who could access that started to skyrocket, especially as the cost of synthetic DNA fell, and more of us learned how to do reverse genetics, or at least got good enough at tissue culture that we could figure out reverse genetics with better and better protocols. So probably around 30,000 people in the world can assemble any influenza virus. And it doesn't really cost very much, less than $3,000 for the synthetic DNA. Most of us have access to labs. We could do it. Doesn't mean that that virus would take off and cause a pandemic. And in fact, we'll talk about why that's protective. Other kinds of viruses are harder to make, but many virologists are pretty gung-ho about the technologies and say that reverse genetics is available for virtually all families. It's like a Lego set. I wouldn't go that far. This is not easy. Tacit knowledge is a thing. Nevertheless, access is becoming democratized. So there's a lot of bad people in history, right? Think about the Unabomber. I don't know how many of you read the Unabomber manifesto, but his philosophy was basically that technology was going to degrade human dignity because we would make a world much different from the one in which we evolved. That would make us miserable. We would engineer our own children to meet the demands of the market and thereby debase ourselves. And we'd be better off if we just crashed civilization and lived as hunter-gatherers. So he had a particular beef with biotechnology. He wanted us to stop technology development and get rid of all technology if at all possible. And this guy was a Berkeley mathematics professor before he went to live in a cabin in the woods and murder people with mail bombs. He was good. What if he knew that he could have deliberately caused a pandemic? Would he have done that to discredit biotechnology? Maybe. We also know that the lead biological weapons designer for the apocalyptic cult Aum Shinrikyo around 1990 was a graduate-trained virologist, and he wanted to get his hands on Ebola to use it as a weapon against civilians. Aum Shinrikyo wanted to kill as many people as possible to bring about the apocalypse. And they had a sizable budget. But in 1990, thankfully, none of these technologies existed. So that's a couple of people, but no one has made a pandemic deliberately. That's good, because if they did, I think there would be quite the backlash. We've already seen a loss of trust in science over COVID, very unfortunately. And if there was a deliberate pandemic, we would really not be allowed to do science freely anymore, anywhere near the way that we are now. So I'm incredibly interested in preserving our ability to build a flourishing future with biotechnology, which means we need to figure out how to ensure that we don't ever see anything like a deliberate pandemic. And I submit the reason why we haven't seen it so far is actually we don't know which viruses are exceptionally likely to cause them. Obviously, we know that anything we find, by default, we sequence it and we put the genome online. Synthetic DNA, the cost is thankfully falling because it's the backbone of biotechnology. And as mentioned, a lot of us can turn them into pandemic viruses. Now, if you Google for which viruses are likely to cause pandemics, you will get the most ridiculous amount of WHO articles talking about spillover risks. And none of those viruses is going to cause a new pandemic. If you ask an LLM in the right way, it will give you some better advice. It will sort the top ones. The top ones will rise to the top of the pile. And it'll suggest ways that you could obtain those. And it will do horrible things like recommend that you infect sleeping people in airports in order to cause a maximally fast-spreading pandemic with whatever virus you can get. That's an example of how LLMs could potentially give people knowledge of how to cause harm that they couldn't find otherwise. But we still don't have any great ideas as to which particular viruses are really, really likely to cause pandemics. What really worries me is generative AI applied to viral entry proteins. So here's the thing. We all want to know which particular mutations are likely to occur in an entry protein we're targeting with a vaccine or an antibody. Because if we know that, then we can redesign our therapeutic in order to make it harder for evolution to escape it. The problem is, if you create a general purpose model that is trained to predict the effectiveness of viral entry for a given sequence and the extent to which that sequence will evade existing population immunity, then you have a tool that is a predictor of whether or not a virus equipped with that viral entry protein will spread widely in humans. That is, after all, the point, right? You want to know which mutations would evade immunity and still be transmissible enough to spread widely. That's what we want to block with our therapeutic. But you cannot acquire that knowledge without also learning that you could make that particular variant and have a good chance of causing a pandemic. Now, the model itself is harmless until someone demonstrates that it's credible, that it actually works, that 1 in n sequences actually would spread widely and cause a new pandemic. Would it be a bad pandemic? Probably not. After all, we would still have T-cell immunity to the parent virus, and you only would have changed the entry protein. But it would be bad. And it would be bad because a lot of people could do this. And if a state actor wanted to do this, well, if anything that's accessible to terrorists is something that can be blamed on terrorists. Now, what could we do about this? I mentioned that vaccines are slow. Well, our current national biodefense strategy, which sadly has not been fully funded, has 2 goals. It's vaccine focused. It wants to even improve upon the miracle that was Operation Warp Speed and make a new vaccine within 100 days of first getting the genome sequence of the virus. Now, obviously, we can design it in 1 day, but they want to get it all the way through clinical testing and production. Approval in 100 days. And then 1 month after that, they want enough doses for all Americans. But note, that's enough doses. That's not actual shots in arms. And if the vaccine requires multiple doses, we're in trouble. And if it's not very effective, we're in trouble. But thus the problems. Vaccines are great against natural pandemics, but deliberate ones are going to be faster than natural, and natural is still a little bit too fast for us. So the Omicron variant spread from somewhere in southern Africa. We detected it, got the first sequence, and within 100 days, it had spread to the other side of the world and infected a quarter of Americans. And by that point, it had infected half of Europeans, so on other continents. That's fast. But if the first time we ever saw it, introduced in an airport and then spread immediately by air travel, that would be all over the world much more quickly. So that would be bad. And if you have a tool that will predict how to cause lots of pandemics by making, of course, many different variants of that viral entry protein, then they could make several at once. And then after a few months, after we frantically try to develop vaccines, they could do it again unless we caught them. And every vaccine costs us around $17 billion to make and distribute. So it's offense dominant also in the sense that it costs us much, 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 much more to defend against it than it costs one person who decides that they want to cause large-scale harm. And of course, if biology is not just offense dominant within biology, and we'll talk about better defenses later, but it's also hardware insecure, then our vaccines may not be reliable. So we might want to assume that, infection might be death. We might not be able to make a vaccine if it's deliberately engineered. So really what we want to do is ensure that people can't get DNA that could cause a pandemic unless they're a legitimate researcher doing awesome research to save lives. How can we do that? Well, the way most of us think about when it comes to screening DNA is you want to search for the best match. You want to know, is this DNA sequence order something that is a problem? So what's the best match? Let's just blast it. Now, the problem is BLAST was not designed to detect whether or not a sequence is dangerous. It's a tool for detecting taxonomic relationships. And you can imagine that if you want to distinguish between, say, 1918 influenza, potential pandemic pathogen, and a regular seasonal influenza strain, that could be difficult because they're both influenza A. And that means that they share a lot of sequences. Now, if you are looking for a long enough sequence, the odds of having a 200 base pair chunk of any given influenza virus matching another influenza virus is pretty low. So you have near 100% specificity. But if you want to detect smaller pieces, and remember that we can assemble whole genes, whole viral genomes from 60 mers pretty readily, then you can't just screen 200 mers. You need to screen shorter sequences. And then your specificity drops. The odds that any random flu strain will share a 50 base pair segment with 1918 is roughly 50%. And that's also true of all of the other controlled strains. So it's just really hard to use BLAST to distinguish because you have to check every window of every oligonucleotide or gene that you're synthesizing and see what the best match is. And a lot of those are going to be identical to ones that are in controlled pathogens. So what do we do with this? Well, maybe we should go back to tools like Kraken and do exact match screening. So you can imagine that if we define controlled sequences as being this beta region here, but we also need, and then we can define alpha as the set of known unregulated sequences that can't cause problems. We want to let everybody get alpha and not beta. But we also probably need to screen for functional variants of beta. So how can we prevent people from getting the virus, but of 1918 virus, while still allowing them to get less harmful flu strains for legitimate research? Well, in principle, if we're just compiled the set of fragments for the agents that should be in alpha and in beta, some of them are going to be shared, this region in both, but we want to look for the ones that are unique to beta, both predicted and not. And if we only screen for those, then we can ensure that anyone who needs those sequences to make the viral backbone, which is not going to change, I think, with generative AI, going to be very hard to evade. then we can reliably distinguish between sequence orders that are dangerous and ones that are safe. How well does this work? Well, again, influenza is the hardest test case. And it is admittedly hard with more recent strains of influenza, where you really only have about 2% of your 30mers that are utterly unique to that particular year's strain. But for 1918, you're up to nearly 20% when you still have effectively perfect specificity among all known influenza strains. And even 2% is still better than nothing. So you're in, and this is the worst case scenario. Influenza is the hardest to distinguish. Every other potential pandemic pathogen is much more unique relative to all other viruses that we've found than are the influenzas. So we have a pretty good chance of distinguishing, even at the 30mer level, between controlled things and less dangerous things. But of course, I mentioned earlier, what if someone just includes mutations and designs them to not impede the function of the virus? Well, in principle, there's nothing stopping us from computing those predicted functional variants in advance and then checking to make sure that they don't match anything that is known and unregulated, any harmless sequences. That way, we can detect not just the sequences that are unique to the known agents, we can also predict potential sequences that are distinct and could be substituted and screen for those as well, while retaining our specificity. And this is just really, again, hard to do with BLAST because your specificity is so low for short sequences as is. So what is our vision? Well, ultimately, we can order DNA from a synthesis house and have it come in the mail. Or some of us, particularly chemical biologists, have benchtop synthesizers. In the lab when I was doing my PhD with David Liu, we had a benchtop, and I would sometimes spend time over the weekends making primers for the next Monday because I was impatient. Terrible idea, never do that. But what we need to do is screen both of those. And we need to ensure that legitimate scientists trying to save lives and cure people can get whatever known safe sequences they want and can easily get controlled dangerous sequences with an authorization check to verify that they're legitimate, which again is something that we can effectively automate. We can, for example, turn your biosafety permissions that you get from your biosafety officer saying, yes, you have the right to work with this gene, this virus, etc., into a PKI certificate that you can upload with your order, and then it will allow you to order anything that you are deemed safe enough to work with. We need to work this into not just the synthesis houses, but also the benchtop synthesizers to prevent people who are not legitimate and might want to misuse these capabilities. How do we do that? Well, here's where I'm going to borrow some slides from an org that I co-founded a while back. It's based on a technology that we developed to do this kind of thing. So it's now an independent Switzerland-based nonprofit, and they offer synthesis screening for free using this technology. But if any of you happen to work for pharma, you might be saying, whoa, 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 we can't just have everyone look at our gene synthesis orders. Those are trade secrets. You need it to be privacy preserving. Well, we'll get there. What we really need to know is this robust to attempts to evade screening? We really need to check this in the lab. So to find out, we said, okay, the way this screening system works is we're looking for specific sequences. And in this case, we looked at peptides, so windows of 19 amino acids in the various proteins of M13 phage, which was our target virus. And we split into two teams. We had a red team designed and launched attacks at each window. So they used computational tools to predict which particular variants of that protein they thought would remain functional and be different enough to evade the screening system. And the blue team then designed the screening system to attempt to detect those. And the red team then actually made phage-mid libraries with these variant proteins and knocked out the native protein in the phage, and then measured the fitness of the mutant protein relative to the wild type for all of these members in the 21,000 members in each library for each of these 9 windows. And then we could compare how many of these attacks were functional enough that the phage, the virus, could still effectively spread, but also evaded the screening. And here's the results if you look at two different windows. Some windows, the one on the left is easily defended. It's highly defensible. It's somewhat more conserved. And so the vast majority of attacks were here on the right, the ones that had high predicted fitness. Those were the ones detected by the screening system because it predicted that their fitness was high. The yellow ones got through. But now note that this is just one window. And if you're trying to evade screening, you need to evade screening at all of the windows. And while the window on the right isn't nearly as effective, you need to, again, evade the one on the left, the one on the right, and the other 100 or 1,000, depending on how large the sequence being protected is. And the neat thing is, because we actually measured the fitness of these, we could calculate what is the fitness cost of including these mutations, and then using various multiplicative or additive assumptions in terms of the fitness cost, we can then estimate what is the effect on the capacity for exponential spread. That is, how does the mutation change the R0 of the virus? And what does that mean with respect to, could we defend against someone trying to obtain different viruses based on their exhibited R0 when they spread? So measles is extremely contagious, and so it's way down here. So this is so if you could get a yellow dot above the measles line, there's a lot of yellow dots, then that mutation was not so costly to prevent measles from spreading. But it might have been costly enough to prevent, say, the flu or SARS from spreading. And when you multiply it out, essentially it's really, really difficult. But that was done before we had really good generative AI and even structural redesign tools. So we wanted to know, well, what if someone tries to redesign an enzyme? If we treated that enzyme as a hazard, could we still detect it, or would their redesigns that were functional get through? So here's the Baker Labs redesigned TEV protease, which they used protein MPNN, and they got a bunch of their redesigns. Some of them were actually more fit than wild type, which is a fascinating phenomenon that I'm really interested in exploring further. Part of it's because TEV's not very stable in the wild type. But nevertheless, they got quite a bit of sequence identity changed in these enzymes and still were highly functional. But for all of the functional ones, when Secure DNA treated the TEV, the wild type TEV, as a hazard, it detected all of the designs that were functional. Detection started to drop a little bit for the ones that are less functional, and so on. But point is, this detected even redesigned proteins. Never going to detect a de novo protein, but again, you can't de novo design a whole virus. I think that is many, many years away from us. So this suggests that if we can do this with extremely crude, actually partly Blossom 62 based prediction tools, then if we actually used protein MPNN to generate the predicted variants in the database, then that will be even more effective. And Secure DNA was tested on a bunch of actual customer data from a variety of gene synthesis suppliers around the world, total of 67 million base pairs. And without checking for which sequences from viruses were known, we got 1,400 out of these 150,000 screened genes and oligos were hits. That is, they matched some regulated bacterium or some human virus. Again, not actually regulated, because the microbial genes were housekeeping genes or the like, and the human viruses were not regulated human viruses, but we just still wanted to detect them because, again, in future, it may well be possible to turn any given human virus into a pandemic. So we probably want to keep track of those. A small fraction of them were toxin genes that confer pathogenicity to a bacterium. Some of them were actually hazardous regulated viruses, things like, yes, Ebola. And some of them were related viruses that were not actually controlled, but were similar enough to the controlled ones. But if you go through NCBI and you take every existing strain of every virus in NCBI, and you check all of these related viruses against the unregulated ones, and you let all of those through, which is what the system does, then there are just 22 30-mer windows that were remained. And 22 windows were there because they were not known viral sequences. They were almost all influenza strains, but people were ordering either mutants of influenza strains probably through deep mutational scanning or the like, or they were ordering a strain that was not in NCBI. So it all depends on your database. If your database is really good, because you have all known sequences, then only truly novel sequences that have never been reported and put in a database could possibly come up as being controlled when they're not. So your false alarm rate can be as close to zero as your known sequences are. And of course, you want to know when you're placing an order, is it going to be a problem? So it gives you an answer very, very quickly, tells you where is the problem among known controlled sequences. And it does this really fast, faster than the website time of the typical synthesis provider. So you don't have to waste your time placing the order and then being told the next day, oh, this thing happens to match a controlled sequence. Can we talk to your biosafety officer, which nobody wants? You can just tell you immediately. And it doesn't cost very much to run, which is why the foundation can offer the system to providers for free. But what about those trade secrets? So ideally, you want to ensure that the system is privacy preserving. You don't want to have to disclose anything about the sequence you're screening to the screening system unless it's actually a controlled sequence. How do you do that? Well, this is why the bulk of the secure DNA team was cryptographers from around the world, because we wanted to ensure that screening was privacy preserving. How can you do that? Well, the basic idea is use what's called an oblivious hash that is computed by multiple parties. So we cut our orders and our hazards up into these windows, 30 base pairs and typically 19 or 20 amino acids, although we're decreasing that. And you choose which ones you're going to defend at random. So an adversary doesn't know which ones they would need to change in order to escape. They'd have to change them all. We predict the functional variants. We curate, remove all of the known harmless sequences to avoid false alarms. And then we just check the orders against the hazard database. But specifically, we first hash them obliviously. So we turn them into these gibberish strings that can then just be compared to see if they're identical. So how does the cryptography work? Well, you have Claire, the customer, talks to Cynthia, the synthesizer, and Claire wants to give Cynthia the sequence. Cynthia has to know the exact sequence because Cynthia is going to make it. So you can't have privacy here. But then you want the database, we'll call David, needs to check to see whether or not any of those windows within Cynthia's sequence are hazards. And Cynthia needs to know this so that she can say no to Claire if there's a hazard in there. And you can do this, of course, by having Cynthia hash the sequence. But the problem is if you then have to tell David how to do that as well. And then an adversary who's eavesdropping could just do this and essentially take every possible query, hash all of the sequences of that length, and then reveal what the sequences are. So to get around that, we introduce a key server who needs to participate and provide information into the hashing step. I'm skipping over the cryptography here for simplicity, but the basic idea is we actually split the key server Kate, so there's multiple of them. So now the synthesizer then performs a local blinding step. She hashes it and then raises it to a power that only she knows. She then sends this to the key server network, which collectively compute a second hash. And this hash is constructed so that you can then reverse the exponentiation on the synthesizer's side in order to get a sequence that is the same as if you just sent the original sequence to all of the key servers for one-step hashing. So effectively, you can undo this blinding exponentiation step completely. And that means that Cynthia's sequences can be directly compared to David's sequences in the hazard database, which means that nobody needs to know what anybody else has unless two of them happen to match, in which case then you know that that particular sequence is the same thing. And this is nice because in principle, suppose someone builds, wants to build a novel wildfire agent. Well, if someone anticipated how you could engineer a virus to be much worse, to spread like a wildfire, and then in principle, we could screen for that without having to tell anyone that that's what we're screening for. I'm going to skip the next bit. But this in principle would let us screen for things without disclosing what it is that we're worried about. It's unclear whether or not we'll ever actually get there, but for now it gives us some nice privacy preserving properties. And this is also important because screening needs to be an international endeavor. Pandemics don't know any borders, which means we need all nations that synthesize DNA to screen, and we need them to talk to each other, because we ran a little experiment in which we, with FBI awareness, checked to see whether folks would send us, or rather send a person using a pseudonym, sequences from 1918 influenza. So about 450 base pair chunks, and ordered from a wide variety of gene synthesis providers. Also ordered pieces of ricin, which is also a select agent toxin, and most of them sent it. And this isn't because some of them didn't detect it. It's because lots of us order individual genes from hazardous things for perfectly legitimate research. And this happens so often that the providers, who have no way of knowing whether or not you ordered all of the other pieces of the virus, tend to just let one or two go through. They don't check. And this is a problem because obviously it's a way of getting DNA through screening. So we really need all of the synthesis providers in different countries to be able to talk to each other, or effectively keep track of which pieces of, say, 1918 influenza people have been ordering, so that we can know to start advising a provider, hey, looks like someone else has been trying to collect all of the pieces for this virus recently. You should start rejecting those small fragments. That's what we're going to need in order to stay safe. And that means the world needs to work together, which is why the Secure DNA team has been international from the beginning, with representation from the US, Europe, and China. So what does all this have to do with generative AI, other than that we need to ensure that nothing goes wrong so that we can continue to bring about that flourishing future? Well, I think it's conceptually very similar to this question of, what do we do to find out whether or not our generative AI models will describe how to make biology more destructive than it would be otherwise? Now, this is a challenge because, just like the case of the screening, where you don't necessarily want to let others know what the thing is, what the dangerous sequence you're looking for might be, because if they know, then they might be able to get around it, or they might be able to misuse it, or what have you. We don't really want to attempt to teach our models what is and is not dangerous, because if they know, then they can generate that dangerous thing. And we can't even share what you might call a grading rubric to check whether a model can disclose how to make a given virus extremely nasty in a creative way, because then anyone who gets their hands on that grading rubric would learn how to make that virus nastier. Now, yes, I am assuming that you can, in fact, engineer viruses to be nastier than nature can. I am biased because CRISPR-based gene drive told us that we can, in fact, build things that are more fit than what nature does. And I fully expect that we can do this directly proportional to our ability to explore the fitness landscape of proteins in ways that we understand. And I think viruses are one of those that we're going to understand well. And I think there's a lot of ways of causing harm that nature doesn't use, because nature optimizes for replication, not harm. So if we want to check to see whether our models do nasty things, we need to have ignorant subjects access a helpful model to ask it how to do nasty things. We need to get the output of that, the transcript of that session, and we then need to grade them to see whether or not any of the nasty ideas that people have had are present in that transcript. And if so, then we can contact the model developers and say, hey, the model is concerning in this way. You need to institute safeguards. How do you do that safely? Well, I think Secure DNA is a model for how this could be done. So you don't want to actually have ignorant human subjects asking how to cause horrific harm with biology. What you can do is you can have an LLM play the role of a human test subject that explores many, many, many conversational paths with a new model. And then we need another LLM, which can be a grader, which can be a trained model that's been trained specifically to evaluate outputs according to this expert provided rubric. And I think the way to go here is actually to have the experts train the model to recognize the thing that they're worried about, whatever that might be. And then that model, the grader, can run on secure hardware that has output keys and master private keys. And then if the grader finds a match, then the expert can be called in to confirm. And then the model developers can be informed that their model is doing this nasty thing. And then we can ensure that our models hopefully do not do those nasty things, because we really don't want to live in a world where you can just ask ChatGPT how to make, I don't know, mumps much nastier than it currently is and evade the vaccine. So that's how I hope we can build that flourishing future without seeing anything go wrong. And thanks to everyone in my lab, most of whom don't do anything in this security general space, but some of which do really amazing work in a wide variety of different areas. And thanks to our many funders. And thank you for your attention. Happy to take questions.
Thank you very much, Kevin. This was awesome. I see many people clapping. Thank you very much, really. We open the floor for questions now. Yes, Chris, please go ahead.
Hey, Kevin, great talk. Awesome. How do you prevent people from getting synthesizers that don't have that protection? Seems like it might become easier to create these benchtop synthesizers, or there might be kind of markets for those that people could get.
Yeah, you're absolutely right that the existing synthesizers are a huge problem because none of the current ones have any kind of screening built into them. So if you have one and you can get the phosphoramidites and you know how to assemble those oligos into gene-length things, as many people do, then you can still get potentially pandemic-capable viruses if you know what one is. So what we're going to need to do is launch a trade-in program, because thankfully there are not many synthesizers. There's not that many chem bio labs. And if you have a trade-in program for a new model that is probably enzymatic, typically much better, then if that new model has the screening built in, offer subsidized trade-in, then we can replace all of the insecure ones with secure ones. But that is going to take time. I just hope we get there. That said, security is a matter of Swiss cheese defense. So you want to do what you can with the knowledge that we're not starting from a great place. And if we get almost all DNA is from the synthesis houses, and it's certainly much easier to order all of the segments already pre-cloned into reverse genetics vectors from a synthesis house than it is to make all of the 200 mer oligos on your synthesizer and stitch them all together and do it all yourself. So there is a relative bar that affects expertise and access. Great question.
Other questions?
We have a question in chat from Sean.
Yes.
How terrified should we be that an AI-generated biological apocalypse is imminent? I'm not worried about imminent. I mean, still the number of extremely obviously credible pandemic-capable pathogens that we know about is really one, which is smallpox. It's very clear that smallpox would cause a pandemic if someone makes it, but smallpox is 186,000 base pairs. And in terms of AI generated, in terms of the AI tells you how to make a new pandemic capable virus and make it nasty, well, none of them can do that now. That's we and others are monitoring for that capability. And I hope it's many years away, but we'll see. It's a challenge.
So I have a question. What do you see as some of the protective factors? Like what's on the side of helping us prevent such apocalypse?
Most people are good. I mean, you'll note that I had to reach back into history, back into the 80s and 90s to find people who clearly had the motivation and some of whom had the training and talent to potentially do it. And despite we hear about all these bad things in the media, they're actually fantastically rare. We just hear about them because of negativity news bias in the media. So almost everyone is good and would never dream of doing anything like that. And so even if it becomes possible, then probably no one is likely to do it for quite some time. What we're talking about here is something that would be devastating if it were to happen. There's only a very small chance that it actually will, even if the capability becomes reasonably public. It's just, I don't think we can afford to take that chance because we need to preserve public trust. And that's just an ethical imperative. And it's also necessary to build that flourishing future because we can't just build it and leave the rest of humanity behind. We're doing this for everyone. And so we have to bring everyone along. And that means we need public support for the things that we're doing. And if we screw it up, then we could lose a lot more than just the folks who would die. So
I see a question from Zeb in chat, but before that I have a follow-up, which is what can people on this call do to support you?
Oh, that's a great question. Talk about how we should think about this as scientists, right? Because it's not natural to us. No one trains us to think this way. Most of us don't think about how our research can be misused. And that's probably also protective, because you asked what are the protective things. Also, we don't know which viruses can cause pandemics other than smallpox. Do we want to open source models that will predict viral escape variants of vaccines? Because I don't see a way that you can make those models credible for the purpose for which they're intended without giving everyone the ability to make a new viral variant that will spread widely. They seem to be two sides of the same coin. And we need to have a discussion as a community. Are we going to do that kind of thing or are we not? And this is hard because we're not used to thinking about that. But I think we have to do it. So if you can talk to folks and think, should we be doing this? Similarly, mirror life. Should we just decide not to make mirror bacteria because that would be much worse than any pandemic, as best we can tell? Is the risk too great and we just shouldn't go there? It's a matter of just read up on the benefits and the risks and talk about it with folks. And I think we need to decide as a community. So bring up these issues, think about it. Let's not just assume that everything will go well to build that flourishing future. We need to not only do the hard work of building it, but we also need to prevent those low probability things that could just sweep away everything we've built.
Yes, thank you for that, Kevin. Let's get to Zeb's question. Do you want to unmute yourself and ask it, Zeb?
Yeah, I was just wondering, you mentioned that vaccine approval process is 100 days. Do you know of any efforts being made to speed that up, or if AI could help?
Yeah, there's a lot of people working to speed that up. I think there's a lot of potential for AI to speed up clinical trials in various ways, including by doing simple things like organizing volunteer networks. So I don't know if any of you are members of One Day Sooner, volunteer to test vaccines earlier. That's the kind of thing that we need, better organizing of people. But a lot of it is regulatory barriers. Those can potentially be surmounted. A lot of it is just straight up manufacturing time, is my understanding. It's not so much the design of the initial vaccine in most cases. Now, what would be transformative is if we could get actual broad spectrum vaccines to work well. But I know there's many folks on the call who are working on that and know much more about it than I do, so I won't speculate on that. It is certainly true that you can imagine a day when we have desktop bioprinters in our homes and you just download the design for the new vaccine and inject it into yourself. That's a promising future. It's still not one that I'm super happy with because it presumes that the vaccine will always work. And I'm just not confident the vaccines will always work. So I'm not willing to go to a world where we just try to install our biological software updates every day. That just doesn't seem reliable to me, but I could be wrong there. My preferred way to stop pandemics is just to ensure that we actually stockpile protective equipment, because the number one reliable way to stop a pandemic is to not get infected, and specifically to ensure that fewer people get infected than were infected in the previous viral generation so that it goes away. And the worst possible pandemic, something measles level or worse, could be stopped if we had enough, certainly powered air purifying respirators for all essential workers, and then could make more for enough people. That will stop any pandemic. There's a really exciting work on far UVC light, which is germicidal but doesn't harm us. There's also some incredible emerging work on glycols. This is something that was investigated in the 40s before we had the measles and mumps vaccines. They just sprayed triethylene glycol or propylene glycol in the air as misters. Now, these are some of the same chemicals that are used in stage fog. So very common, we know they're safe. They're in fact generally recognized as safe, regulated, and they're germicidal in the air. So we could use those as passive defenses in areas just to suppress the transmission of everything respiratory. So you put these things together, if we just have a stockpile of good enough PPE, and we have a list of all of the truly vital workers, food, water, power, law enforcement, and we can ship it to them, which of course is something that the industry can do, then we're proof against basically all pandemics. So I would just rather that we do that, which would cost less than developing and distributing one vaccine, to just create that stockpile. We would be good from there on. That's what I would love to see, because then we wouldn't have to worry so much if we can reliably defend against pandemics.
Thanks. I think you did talk about that in the podcast as well. I remember we are almost at time, unless you are willing to comment quickly on the question in chat from Gary.
Yeah, so how do you prevent other forms of misuse? So what do you do to control open source Gen AI itself? So here you have the biodesign tools, and I mentioned, in general, I'm not super concerned, except about the ones that are specifically combine function of something like a viral entry protein and immune evasion. Just structural prediction and so forth on its own, not such a problem. But really, those viral entry proteins are the immediate issue. In the longer run, we might have other issues, but those are the big one. My preference would be to say, you know what, let's ensure that everyone who's developing biodesign tools has to register, become part of the developer community. We have share models freely in the developer community, but they're not open source. You have to register in order to get them. And then everyone else in the scientific community who wants to use them operates them through an API. I think that would allow us to check to ensure that people aren't redesigning viral proteins without permission. But even there, I don't know if we'll get there or not. That's just an area where the scientific community needs to talk about it. LLMs, that's a more complicated question. And I think we should probably call it a day or a night, depending on where you're at. Sonali, sorry, you're muted.
Sorry, thanks. I was just thanking you. Thank you so much for your talk. We really enjoyed it. This was amazing. And thank you, everybody, for your questions and for attending. So we'll see you next time. Thank you. Bye.